Thank you. Sue is going to be talking on the exposure to chlorpyrifos-based pesticides, the effects on bees in Central Otago. Sue Michelsen Heath, welcome.
Thank you very much. I'd just like to correct Barry and say I haven't yet got my PhD. I'm working on it. I'd like to tell you today about a very large field project that a multidisciplinary team started in Central Otago. I set out to look at pesticides in bees, and I set up the field program, and once we'd got underway, it was very obvious that it would be an opportunity for other people to work on the same project, and listed there are some chemists and another zoology student, Matt Hayward, who made projects on that same field setup. So we tried to share the information. It's essentially a very large project. It involved a lot of people, a lot of setting up. You know as well as I do that bees are subject to a great cocktail of chemical effects. They're either ones that you introduce into the hive yourself by treating for varroa, or they are ambient in the atmosphere or in the soil or in the environment around your bees because of things like spraying and other processes. I want to introduce you to a nasty chemical, and I want you also to remember a very fine lady who it continues to amaze me nowadays, and that was Rachel Carson. I reread her book Silent Spring a while ago, and in 1960s and 50s she talked about chlorpyrifos, and she told us that there would be problems and that they would be for flying insects and pollinators. And chlorpyrifos is an organophosphate, and she identified then the dangers of this type of substance. They're very old generation pesticides. They're not ones we've produced recently. They were first introduced by Dow Chemicals around about the 60s, but had been evolved and worked on during the war and were actually used as a neurotoxin. We know that internationally it's used a great deal. You may have been following the debates in America at the moment between the yes, we will use it and no, we won't, which is part of the present presidential regime. What we do know is that it's a motor and cognitive effector. It works on the neurotransmission of signals in the animal's body, and it affects humans as well as insects. It will impair things like foraging. We have enough evidence to suggest that it does affect the motor neuron function of bees. There's great concern over its use because it is old worldy. There are other things that may be able to be used instead, and yet it still persists. This is a pretty picture. It doesn't necessarily suggest that the pesticide is directly applied to these fruits, but it is used in the industries which produce these foods, and this is happening in New Zealand. Talking to people over the last few hours, I've discovered there's great variability regionally on where things are applied. There are hot spots, and one of the reasons that we chose to work in Central Otago was because it is a hot spot. We began the study before I began my PhD back in 2016, when we randomly surveyed hives right around the Central Otago area. And I just took foragers from the front door of hives, and we examined those chemically for what sort of content that they had. We looked mostly at exiting foragers because they've already got rid of their waxes and their their nectar, and so we don't have so many things to clog up our chemical experimentation and our analyses in the lab. We took 3 representative hives per aviary. I looked at 35 Central Otago apiaries, and the results from those 105 hives altogether were that chlorpyrifos was found in 17% of apiaries randomly, 12% of all hives, and the concentrations were really quite low but still present, and they went from 35 to 286 picograms, which is 10 to the minus 12 grams, really tiny, but being well below the LD50 level, it's still of concern when it is found without being close contact to many spray areas. Research was done by Elodie Urlacher in our laboratory as a result of my collection of bees, and Elodie tested using odour-associated learning, where you were rewarding animals for recognising particular odours. She found that there were problems with learning odour differentiation and memory when you applied in field-matched levels of concentration. So this implies that there could very well be problems with recognising flowers, being able to differentiate between odours, and remembering what you have been feeding on, and particularly maybe remembering where you were going to when you were going home. So the core questions I wanted to find out about were, if we have exposure to chlorpyrifos sprays in Central Otago, does the chlorpyrifos accumulate in the bees with time? And if you introduce bees to an area that has been exposed to the spray, does it matter when you introduce them, how long after the spray event? And the last questions were, what is the fate of the chlorpyrifos if it does go into the colony? If those forager bees are actually vectors for carrying it in, and what effects will it have? We chose Central Otago because it has agricultural diversity. There's a lot going on. There's all sorts of food crops, animal fodder crops, an amazing array of landscape types, and there's high chlorpyrifos use. I want to show you our field setup, and this might give you some idea of the scale of it. With the generosity of farmers in the area, we were able to set up a very large basin and plant and prepare the soil. We used Phacelia as a crop, and we planted it out, and there is it in full bloom. It's highly attractive to bees. It's nutritionally valuable for the nectar and particularly for the pollen. We put in some beehives beforehand, and we called these the kamikazes because we anticipated that being in full contact with the spray during the spray process, they probably would all die. And we only chose 5 for that reason. We use a boom sprayer, and then after spraying, we introduce colonies, 10 per group, and, oh, sorry, you're not hearing very well. Yes, we introduce 10 sets of hives per time schedule, and you can see them laid out here in groups. They're only showing you 2 lots of 10 there, but we used 60 in all. And we also had a site well away from here, which was our control site that was not in any way sprayed. And what happened was that we introduced, first of all, the group before the spraying, and then we took these lots of 10 in 2 days, 4 days, and 6 days after the spraying event. And we also moved a set of 10 hives to another location to see whether the movement itself had any effect on production. And we kept the control site as closely matched as we could, well away from the area. This gives you a bit of an idea of the schedule. Every one of the dots on the schedule represents a collection of foragers, nurse bees, pupae, pollen, emergent adults, drones, and queens. And so we went on for a whole 3 to 4 months, and we were very acutely aware of the fact that your bees are picking up pesticides, not just by respiration and contact, or through pollen, propolis, or nectar, but 2 other things as well which you might overlook. One is water, which is not so important for bees with chlorpyrifos because it's not a water-based, water-soluble pesticide. But it is also the issue of flight, because we're now getting research results which suggests just the process of moving through an atmosphere which either has volatilized pesticides or direct droplets is that the wings themselves are able to absorb over that large scale area quite a lot of pollutants. So the results of the study showed that the levels in forages alone was well above what we found randomly. We would expect this. We deliberately set it up so that we would get this intake of pesticides so we could see where it was going. So that was not a surprise. But what was interesting is that we've got queens at the top of the ladder. Queens accumulate more pesticide than any other bee in the colony, and that's because they're being fed by others and constantly receiving input from elsewhere. Nurse bees are constantly taking food and passing it to larvae. Foragers had less still, emergent adults less, and the drones least of all. The larvae, having listened to the talk this morning, you could anticipate that with a spray that is soluble in fats, that the larvae would pick up a lot. We found that the pollen was nearly twice the amount of chlorpyrifos per unit of weight to any of the bees or the foragers. And this graph represents, if you think of each of these as a parcel of chlorpyrifos, there's a rise from all of the baseline levels, which were 0. The blacks were the ones that were there all the time. The reds have been introduced later. The greens still later, and finally the blue set were introduced at the end. Whatever the sequence, this is accumulation. So you are getting it into your hives all the time. What we suggest is you replace queens because of this load that they get. I know most people do, but on the other hand, some hang on and hope that they'll go further. The queens are obviously affected by this process. Old comb is waxy. It will have dissolved in it a lot of pesticides and a lot of chemicals. We did a lot of infrared work too to see whether or not over the winter following the exposure to pesticide, whether or not there was a difference in the weights of colonies. And the take-home message from this is that we used controls, those that were only moved but not treated, the kamikaze group that had most exposure, and the remaining group, which are significantly different from some of the controls, but still not greatly affected. But obviously the kamikazes, the ones that were exposed throughout the process, had a problem later, and if you'd like to have a look at my poster, you can see more details. So do check your hives and do consider using infrared monitoring. You can get a picture of your hive colony cluster quickly. You can move through a whole group of hives, and if you've got 500 hives, 1,000 hives or more, and you're working in the winter and want to get an assessment, I can really promote it as an idea. I may not have time to go over this, but what I'm doing at the moment in the laboratory is to culture bees and add pesticide to the culture medium. And I also compared the results with emergent bees that came straight out of the hive to see what percentage normally hatch. And I got quite a good match for that. And what happens is, if you have a look at this, you can see that there is a significant difference between those bees that have had chlorpyrifos and those that have either not had any treatment at all, the second group along, those that came out of the hive, and those that have had solvent only with no dissolved pesticide in it. And finally, when you get along to the end, if you have a high dose, you've got a big drop-off. And considering this aspect, with research that's coming out at the moment about the micronutrients improving immunity in your hives, it does suggest that this is well worth doing. How can we mitigate this? I'm running out of time, but I think this last bit is really worth it. When I put this together, I thought, how can I make some suggestions to you as beekeepers when the problem is so universal? And I think some of my suggestions are fairly naive and basic, but I think we do have to keep reminding ourselves about these factors. It's costly to lose big colonies. It is more costly to lose those than to replace a queen. You need to replace old comb on a rotational, this is the very last page. You need to replace comb because you've got these pollutants still stored in the wax, which obviously are coming back and influencing your hive. You could perhaps use infrared monitoring and even remote monitoring so that you get on top of these problems sooner. I'm suggesting you feed honey wherever possible rather than sugar. And in the peak season, try to avoid treating when you also know that there are spraying going on in the area. And probably above all else, wherever it's possible, could you please have dialogue? We are seeing in Central Otago, just because of our work, that beekeepers and researchers are getting together with spray contractors and saying, how can we resolve the problem? And it's not so much that spray contractors don't do as they're told, but there are some unlicensed spray contractors who go out and do things that are not according to the book. Do consider supplements, and I do stress the dialogue. I have a website which is quite simple to follow there, and on it there is a pesticide survey to see how many people have been affected by it or know about effect on their hives. If you're interested in doing it, please have a look at the website altogether. I'd like to thank particularly any beekeeper, and I know there's one or two in the audience here who helped with this project. It was a massive project, and we got an amazing amount of help from other people and also some sponsors which are listed there. I can't thank you enough. 2 PhDs and 3 masters will have come out of this project, and we're very, very grateful. And I'm terribly sorry I've run over time so much.